Okay, hello everybody, welcome to our new lab. In this lab, I'm planning to talk about another psycholinguistics method which is very common in linguistics studies, which is self-paced reading method. So again, in the first two parts of this lab, I'm going to talk about the theoretical backgrounds of this method, and then in the final part we will learn how to make a self-paced reading sample study in PsychoPy. All right, so let's start. So first, we will be talking about what self-paced reading method is and when should we use it. And then we will talk a little bit about the procedure and algorithm of self-paced reading method in the second part of this lab. And finally, we talk about the ingredients and the PsychoPy file. All right, so self-paced reading method is mainly used to study the reaction times of participants when they read or listen to a part of a sentence. So the idea behind that is that if there is a difficulty in part of a sentence, it will take longer for participants to read that part of the sentence. For example, let's take the following sentence. John said hello to Jack while she was crossing the street. So this sentence, as you can see, is divided into different regions, which are represented with slashes. So John is the first region, said hello to is the second region, so on and so forth. And the way it works is that participants see each region on the screen. For example, they see first John on the screen, but other regions are hidden. And then for each region to appear, they must press a key, which is usually the space bar on the keyboard. They press the key and then they can read the next region. So this is how it works. So first they see John on the screen, and then when they read John, of course they should read it silently, they don't read it aloud. They press space and then said hello to space Jack space while she space was space, and finally crossing the street. So now imagine that we are able to record the reading time of each region in milliseconds. That is the length of time that it takes for them to press the space bar on the keyboard. So pause the video for just a second and think about this question. Which region do you think will take longer for participants to read? You can pause the video, you can go to slides, check the regions, and then come back to see what the answer is. All right, as you might have guessed, region 4 is the weirdest region, because as you can see here, in region 1 and region 3, we have two male antecedents, but in region 4 we have suddenly female pronoun, which we are not expecting to have a female pronoun here. So that's why this region might take longer compared to the normal sentence. John said hello to Jack while he was crossing the street, because in this sentence the pronoun matches the gender of the participants, but in region 4 it seems that there is a kind of mismatch. So in self-paced reading time, if we compare the reading time of region 4 in the first sentence and second sentence, we might be observing a longer or greater reading time in region 4. It's just a matter of milliseconds compared to region 4 in another, normal sentence like the second sentence, which matches our expectations. All right, so, but what is the procedure? How does self-paced reading method works? So first of all, you should build up your trials. So that's, as always, make your trials. And then you should repeat your trials for all conditions. The only thing that should change is the lexicalization of the trial, which we will explain it just in a second. So after, building up your trials, you should repeat the trials for all conditions. So you should know how many conditions you have. For example, in our study, our variables are gender of antecedents at 2 levels, masculine versus feminine, and then the gender of the pronoun at 2 levels, masculine versus feminine. So we have 2 by 2 design or 4 conditions. As a result, we should repeat each trial 4 times. So each trial is associated with a condition, and as I said, the only thing which is different is their lexicalization. So these are our trials, our repeated trials with different words. John said hello to Jack while he was crossing the street. Here you can see that the gender of the antecedents is male or masculine, so it is masculine. Here we have masculine, feminine, and then we have feminine, masculine, and then finally we have feminine. And then by lexicalization, we mean these words: John, Jack, Mary, Sarah. And then for other trials, you should use different verbs, said hello, said goodbye, or whatever. And then instead of crossing the street for next trial, you should say, for example, he or she was shopping, he or she was, talking on the phone, so on and so forth. So different words, but the same structure. All right, so after building up our trials, so it is time consuming because we should repeat this for all our experimental trials. So if we want to have 32 trials, we should make, in this case, 32 trials by 4 conditions or 128 sentences, which is kind of boring and time consuming. Sometimes finding enough number of verbs or enough number of, proper nouns might not be very easy task. But after we have created our sentences, we make an Excel file, which is very important. So the rows in our Excel files are our trials. So each trial goes into the rows. So we have 32 trials, 32 rows, and then each column is one condition. As you can see, we have 4 columns plus 1 index column, so in total 5 columns. We have 4 columns of conditions. So under each column, the relevant sentence goes. For example, here we have condition 1 sentence, condition 2 sentence, 3 and 4. So our 128 sentences will be located in a 32 by 4 column Excel spreadsheet. So after making our Excel spreadsheet, so here you should have different sentences in these columns, but for the matter of space, I didn't put the content of each cell. You should color code the Excel file in a diagonal way. So for example, as you can see, we have orange, we have kind of grayish, brown, yeah, and then we have violet and green. And the reason for color coding the Excel cells is that each color is representative of a list. So we have the orange colors make up list 1, then the grayish brown list 2, violet list 3, and then green list 4. In other words, you need to make a separate list for each condition. So in this study, we have 4 conditions. You need 4 lists. Sometimes if you have 2 by 2 by 2 design, you need to have 8 lists. That's why for more complex studies with greater number of variables, making lists are very time consuming and prone to human errors. All right, so and we should copy and paste this cell into a different Excel spreadsheet just to have our different lists ready. And now you mix your filler items and main trials in a pseudo-randomized way. If you don't know about pseudo-randomizing your list, you can refer to my previous lectures. And now you should divide each trial in your list into regions. So in our study, we had 6 regions, so we should divide each trial into 6 regions. And now everything is ready to make your input file for each list and feed it into the PsychoPy file. So just a deep thinking question. Think about the CSV or Excel input file which we are going to feed into PsychoPy. In your idea, what columns should it have? Think about it, and then in the next video we will talk about it. Thanks.